Hey everybody, thanks for tuning in to listen to this presentation. My name is Eric Queathem and I'm an undergraduate here at the University of Missouri studying biochemistry and nutrition exercise physiology. The title of my presentation is Beta-3 Adrenergic Receptor Activation Induces Estrogen Receptor Beta and Improves Mitochondrial Metabolism in Adipocytes. Basically all that means is that we have a drug that can activate the beta-3 adrenergic receptor. That causes estrogen receptor beta to be induced and then ultimately facilitates an increase in mitochondrial metabolism for adipocytes, which ultimately will improve health. So before we get too far into that, I'll give you a quick overview of what this presentation is going to be about. We're first going to talk about adipose tissue, specifically what happens when adipose tissue is not functioning correctly, and the role that estrogen and UCP1 have within protecting the adipocyte. I'll talk about my research aims and the hypothesis of the study, and then show you guys the hypothetical mechanism that I have designed that kind of explains how all these different components are working together. I'll talk very briefly about the methods we use with this study, and then the results that we have, both the in vivo and in vitro results. And then finally, the conclusions we've made from this study, the future directions we're going to take, and the implications that this research has within medicine today. So let's talk about adipose tissue, or fat. Everyone thinks of fat as being really bad, and everyone just thinks of it as simply storing energy, but it does so much more than just store fat. And there's a really big difference between having healthy fat and unhealthy fat. Whenever adipocytes become dysfunctional, that's a lot of times when we see these types of diseases, like diabetes, cardiovascular disease, even cancer start to develop. And our lab is called the Fit Fat Lab Group. This is what that acronym stands for. And basically what we study is what makes fat healthy and what makes it unhealthy. So one of the parameters that we use to assess how healthy a adipocyte is the insulin resistance. So here is what I mean by insulin resistance. Normally, whenever you eat a meal, here's your blood. Your concentration of glucose in your blood increases. That tells your pancreas to release insulin, and what insulin does is it triggers this GLUT4 transporter to be inserted into the surface of your adipocyte. What that does is it allows the adipocyte to soak up all that glucose out of your bloodstream and thus bring your glucose levels into a normal homeostatic range. The problem, though, is that whenever adipocytes become dysfunctional, they develop what's referred to as insulin resistance. In other words, you eat something, your blood sugar gets high, your pancreas releases insulin, but your adipocyte stops responding to it. And if you can't get any GLUT4s into the surface of your adipocyte, your blood sugar is going to remain high. Most people that are pre-diabetic are usually diagnosed as insulin resistance, and as this becomes a chronic issue, they develop into full-blown type 2 diabetes. And insulin resistance is going to be one of the major markers I use to determine how dysfunctional an adipocyte is. So here's an example of that. One of the roles that estrogen plays specifically in adipose tissue is that it protects the adipocyte from developing insulin resistance. Everyone just thinks of estrogen as a female sex hormone, but it does a lot more than just that. So what we can see, one way you can induce insulin resistance is you can feed a mouse a high-fat diet. What we see here is in this wild type group, meaning a normal mouse, you put them on a high-fat diet and you typically don't see that much insulin resistance develop. However, whenever you knock out estrogen receptor alpha, what you get is a dramatic increase in the insulin resistance of an adipocyte. So before we get too far into this, I should say that estrogen is a hormone and it primarily works through two different receptors, estrogen receptor alpha and estrogen receptor beta. So clearly from this, we can see that ER alpha protects the adipocyte. Whenever you get rid of ER alpha or knock it out, that causes high-fat diet-induced insulin resistance. There's been a lot of stuff that's known about ER alpha, and there's been a lot of studies that looked at it, but there's much, much less known about ER beta. And that's really what the primary focus of this research is what role does ER beta play in the adipocyte? One other thing I want to talk about is UCP1. And what we have found is this drug that we've been talking about, this beta-3 adrenergic receptor agonist, induces UCP1. UCP1 is a really important mitochondrial protein, and it's been shown that if you can increase UCP1 in an adipocyte, that it rescues adipocyte dysfunction. So if you remember on that last slide, whenever you knock out ER alpha, put on a high-fat diet, it develops insulin resistance. That's this one right here. However, if you treat that same mouse with this compound CL, the insulin resistance goes down completely. It completely rescues it and returns it back to what it would be like in the wild-type mouse. So, one interesting thing that we found is that if you look at this figure right here, this estrogen receptor alpha knockout mouse is way more susceptible to UCP1 induction than the wild-type mouse is. This possibly could be due to increased estrogen receptor beta signaling. The reason why is because in this estrogen receptor alpha knockout mouse, you still have the same amount of estrogen circulating. If we get rid of ER alpha, all of that's kind of funneled down into ER beta. And so, if ER beta is playing a role in how CL works, that would explain why this knockout group is so much more responsive to CL than the wild type group is. So what exactly does ER beta do in the adipocyte? Once again, there's not very much known about this. One thing that is known is it protects the adipocyte from Ovex-induced obesity. So unfortunately for a lot of the women out there, you know that whenever you go through menopause, most women tend to put on weight. The reason why is because estrogen really promotes the burning of fat rather than the storing of it. So what we see here is that whenever you Ovex a mouse, if you Ovex a mouse and you get rid of ER beta, it causes the adipocytes to become obese. If you Ovex a mouse and you get rid of ER alpha, meaning you kind of increase the signaling through ER beta, it protects the adipocyte from becoming obese. Additionally, we saw in the same study where we knocked out ER alpha and then hit the mice with this CL drug that CL increases ER beta expression, meaning that ER beta is probably playing a role in how CL works. The mechanism of this is unknown, and once again, that's really what our focus is, to determine is ER beta really playing a role in how CL works in the adipocyte. So this brings me to the major aim and the hypothesis of our study. What we want to do is determine the role of ER beta within CL-mediated induction of UCP1. The hypothesis we have is that if we stimulate an adipocyte either with exercise or this drug CL, that we'll see an increase in UCP1, ER beta, and GRP75. Now this GRP75 is the one thing I haven't talked about up until now. We found out about this, I should say, this is a mitochondrial transporter protein, and we found about it from this paper that's cited down here that basically showed in breast cancer cells, GRP75 can recruit ER beta to the mitochondria. And whenever that occurs, it improves the metabolism of cancer cells. It, in a sense, cures breast cancer. So we wanted to determine, does GRP75 do something similar in the adipocyte? And here is that hypothetical mechanism that I was talking about earlier. So this is kind of what ties together everything. I know this looks kind of jumbled, but I'll sort of break this down for you a little bit. Ultimately, here's what our drug is, CL. It increases lipolysis in the adipocyte. What that means is it's going to take these stored triglycerides in an adipocyte and break them down into free fatty acids. Now, for those of you that don't know, free fatty acids are typically toxic to a cell, and so something has to be done to deal with them. The easiest thing to do with them is simply to burn them or oxidize them, which is something that requires the oxidative phosphorylation complexes. So what we believe is that whenever you increase free fatty acids by activating this receptor, that it's going to activate GRP75. GRP75 binds ER beta in the cytosol, moves it to the mitochondria, where it binds to the mitochondrial DNA and increases transcription of the oxidative phosphorylation complexes. Now, whenever OXPHOS is ramped up, you're also going to see an increase in reactive oxygen species, or ROS. You guys have also probably heard of these called free radicals. which do some pretty nasty things to a cell. One thing that has been shown to sort of buffer the production of ROS is UCP1. So through a mechanism that we're not really sure about, we believe that ROS triggers the nuclear transcription and then translation of UCP1. So up until this point, I've shown you how this drug CL can hypothetically increase GRP75 and UCP1. The last part of our hypothesis is how it increases ER beta. I've already talked about how estrogen is this really important hormone, and it does a lot of other things than just control female sexual development. So the concentration of it in the cytosol is really important for the cell. And we believe that as GRP75 sequesters more and more of this ER beta into the mitochondria, there therefore is less of it in the cytosol. And that then sends a signal to the nucleus saying we need to transcribe and translate more of this ER beta protein, thus facilitating the increase in ER beta, GRP75, and UCP1. Now this is sort of the research methods that we're going to use to determine whether that mechanism really is occurring. Basically what we do is we take a mouse and we stimulate lipolysis by either having it exercise or treating it with this drug. We then harvest adipose tissue from there, and we'll take fresh adipocytes and do Ouroboros. What that does is it's going to measure real time how much oxygen those adipocytes are consuming. As you burn fat and as the oxidative phosphorylation complexes are working, they have to consume oxygen. So that's going to give us an idea of how active the mitochondria are in those adipocytes. Additionally, we can freeze that adipose tissue and later on isolate and measure protein and RNA within them by Western blot or PCR respectively, and that's how we're going to determine how much ER beta, UCP1, or GRP75 is present. The big contribution that I made to this is the in vitro part, basically where we're going to actually isolate pre-adipocytes from these mice. I culture them, differentiate them into mature adipocytes, and then treat them with this drug. And so this allows us to manipulate these cells in a slightly more controlled way than if you're going to do these in vivo in a mouse. So now we'll move on. I want to talk a little bit about this model before we get into the actual data. So I've talked about an estrogen receptor alpha knockout mouse. That is a full body knockout, basically where we completely get rid of estrogen receptor alpha and you get this really dysfunctional phenotype. What we did for this study is we just knocked out this one domain of ER beta, the DNA binding domain. And the reason why is we wanted to see if we got rid of this genomic effect of estrogen receptor beta, what effect does that have on the adipocyte metabolism? We've still left intact this protein interaction domain in ER beta, which is how it interacts with GRP75. And so we're basically just looking at when you wipe out the direct genomic effects of ER beta, what effect does that have on the metabolism of an adipocyte? So now we'll get into the really exciting stuff, the actual data. So if you remember, our hypothesis was that CL was going to increase ER beta, UCP1, and GRP75, and this supports that. What this shows to the figure on the left is that CL increases the concentration of UCP1, and it increases the concentration of estrogen receptor beta. This little star indicates that this was statistically significant, and this circle up here indicates that it wasn't statistically significant, but there was a trend. The reason for this is that we had a pretty small N. There was only 3 per group, so we didn't have a lot of statistical power. But what I think is really interesting is if you look at the fold change for ER beta, we ramped up ER beta 4-fold in these cells. And to me, that's pretty impressive. So this is in support of our hypothesis that CL increases both UCP1 and ER beta. Furthermore, in support of our hypothesis, we show that in vivo, CL increases GRP75 messenger RNA and protein. So for those of you that don't know, a gene is this segment of DNA that encodes a protein. For that information to ultimately become a protein, you have to go through this messenger RNA sort of transient before you get to a protein. That's kind of what the pathway looks like here. And we show that In vivo, meaning in live mice, CL increases both GRP75 messenger RNA and protein expression, which once again is in support of our hypothesis, and the effects of CL on GRP75 are statistically significant in all groups. The last major piece of data that I'll show you is the Ouroboros data, or the oxygen consumption. So to kind of orient you, the left-hand axis is looking at oxygen consumption, and basically these 5 different groups are just showing whenever you add different substrates to the oxidative phosphorylation complexes, how active they are. So the big things to point out is you'll notice there are some diamonds above these groups. That indicates that there's a statistical significance with the genotype. What that basically means is we're comparing the wild type mice that have, they're just normal, and then the mice where we've knocked out this estrogen receptor beta DNA binding domain. And what we see is that whenever you knock out this estrogen receptor beta DNA binding domain, they have suppressed mitochondrial oxygen consumption. That is a huge thing that supports our hypothesis, meaning that ER beta plays a pretty important role in the mitochondria, and when you get rid of this DNA binding domain, the mitochondria aren't functioning as well. What's even cooler than that is the fact that whenever you treat these cells or these mice with this drug CL, you increase mitochondrial respiration through the roof. The stars above all of these groups means that there's a statistical significance for treatment. Basically meaning that in all of these groups, when you administer CL, you increase the amount of oxygen that's consumed by mitochondria ridiculously. And essentially, you rescue the mitochondrial activity. And another interesting thing that we found out is that the estrogen receptor beta DNA binding domain knockout mice were actually more susceptible to this effects of CL than the wild type mice were. There's a couple of different hypotheses as to why that is. I'm not going to get into that, but that's really what's going to be driving a lot of our future studies, which I'll talk about here in just a second. So in conclusion, we showed for the very first time in vitro that CL increases the expression of estrogen receptor beta. We also showed that it increases the expression of UCP1 and GRP75, and it's capable of increasing mitochondrial oxygen consumption. And additionally, we showed that estrogen receptor beta DNA binding domain knockout mice had suppressed oxygen consumption but were more sensitive to CL treatment. So the major conclusion that we can make from this experiment and this study is that estrogen receptor beta plays a significant role in the mitochondria of adipocytes, and estrogen receptor beta may be required for optimal CL-mediated induction of UCP1. So going off of this, the future aims of our lab are going to be investigating the alternative ER beta splice forms, which that's sort of one of the reasons why the knockout mice may be more susceptible. Could be whenever we knock out this DNA binding domain that we get an alternative splice form that has a more pronounced protein interaction domain. And if you remember back to that slide on the ER beta gene, protein, and that hypothetical model, that protein domain is how ER beta interacts with GRP75. So that could explain why our knockout mice are so more susceptible, is because they're able to interact with GRP75 much better. Other aims that we're going to investigate in the future is subcellular localization of ER beta in response to CL. In other words, where does ER beta go when you treat it with CL? We would expect to see that it goes to the mitochondria. And then we'll also study the effects of CL if we wipe out ER beta fully, meaning we do a full body knockout, not just this DNA binding domain knockout, and what happens when we knock out GRP75. So the very last thing I want to talk about is the implications of this research. So given the robust effects of CL on the adipocyte, this mechanism really represents a critical place within current medicine, where we could intervene pharmacologically to treat a wide range of diseases, such as type 2 diabetes, cardiometabolic disease, certain forms of cancer. And I think that in the future, this is going to become a lot more important in how we treat these diseases. Additionally, because so many of the feminizing effects of estrogen are mediated through ER alpha and not ER beta, this really opens up a lot of the different groups that we could use this treatment for. So obviously this can be used for postmenopausal women. This could also be used for metabolically dysfunctional men. We could give this to men without causing these feminizing effects that estrogen normally has. And the last thing is that a lot of times, especially with hormone replacement therapy, that puts you at a slightly higher risk of developing certain kinds of breast cancer. ER beta does not show any of the same pro-growth effects of ER alpha. So by targeting this specific estrogen receptor and possibly combining it with CL and getting a synergistic effect, we could treat these diseases way more effectively than we ever could before without increasing the risk of developing certain types of cancer. So this really is a pretty exciting area of research that I hope everyone's kind of a little bit more interested in from this. So thank you very much for taking the time to listen to this presentation. I just realized that it's about 20 minutes long, and I apologize for how long that is. If you guys have any questions, please feel free to reach out to me. My email's here. I'd be more than glad to talk to you guys, answer any questions you have. I'm very passionate about this research, so I hope you guys enjoyed this and learned something from it. Thank you.